Welcome to Radio Kanchi 90.4 FM हम सबका रेडियो. This is Dr. Soni Kumari, Assistant Professor, Department of Botany, PPK College, Bundu. In my previous lecture, I have discussed about cell cycle and cell division on amitosis and mitosis. In this lecture, I am going to explain about meiosis with its significance. Meiosis is a specialized type of cell division which occur in diploid reproductive cell and form haploid gametes. August Weismann in 1887 predicted the number of chromosomes must be reduced by one half during gamete formation. Van Benden demonstrated reduction division in 1887. The term meiosis was introduced by Frame and Moore in 1905. Types of meiosis. Number 1. Zygotic meiosis. It occurs in lower plant which having haplontic life cycle. Number 2. Gametic meiosis. It takes place in animal mainly for gametogenesis. Number 3. Sporic meiosis. It takes place in higher plants mainly which having diplohaplontic life cycle. Like other cell division, meiosis completed in two steps, karyokinesis and cytokinesis. Karyokinesis involves in two successive divisions, meiosis 1 and meiosis 2. Meiosis 1 is the division chromosomes become genetically different and their number is reduced to half. Therefore, meiosis 1 is also called as reduction division or heterotypic division. Meiosis 1 studied under four phases: Prophase 1, Metaphase 1, Anaphase 1 and Telophase 1. Number 1. Prophase 1. The prophase of meiosis 1 is more elaborate and lasts longer than the prophase of mitosis. It may be divided into 5 substages: leptotene, zygotene, pachytene, diplotene and dikinesis. First phase of prophase 1 is leptotene or leptonema. Leptos means slender, tinea band. In this phase, chromosomes appear as long, thin, slender threads. Chromosome attach its both of ends to nuclear envelope called attachment plate. Chromosome bears numerous beads like chromomeres. Next to leptotene is zygotene or zygonema. Zygon means type, tinea band. In this phase, homologous chromosomes pair together, one chromosome in each pair from maternal and other is from paternal side. Pairing of homologous chromosome is known as synapsis, and during synapsis, ladder-like structure or complex formed called synaptonemal complex. The paired chromosome is called bivalence. Synapsis is of three types. Number 1. Procentric, from centromere to end. Second is proterminal, from end to centromere. Third is intermediate, at various places between centromere and end. Next to zygotene, the stage is pachytene or pachinema. Pachys means thick, tinea band. The bivalent become thick and short, contain 4 chromatids referred as tetrad. Chromatids of same chromosome called sister chromatids. Chromatids of different chromosome is called non-sister chromatids. Exchange of chromosome segment between non-sister chromatids is occurred. Exchange and recombination of segment is called crossing over. The point where chromosome exchange their gene called recombination nodule. Next to packetine, the fourth stage is diplotin. Diplos means double, tenia band. The homologous chromosome starts separating except in the region of crossing over. The point of attachment between homologous chromosome after partial dissolution are called chiasmata. The last stage of prophase 1 is dikinesis. Dia through kinesis movement. At the end of diplotin stage, chiasmata starts to disappear by sliding towards the end of chromosome called terminalization. In dikinesis, exchange of chromosomal segment is completed. Nuclear membrane and nucleolus are disappear. The next phase is metaphase 1. The nuclear membrane and nucleolus are disappear. Spindle fiber are arises from the pole. Spindle fiber become prominent and get connected to the centromere of the homologous chromosome. The bivalent align on the equatorial plane. In this phase, the number and morphology of chromosome can be studied well. After metaphase 1, the next phase is anaphase 1. In this phase, the two chromosomes of each bivalent with chromatids still attached to the centromere separate from each other and move to the opposite pole of the cell. Thus, only one chromosome of each homologous pair reach each pole. Consequently, at each pole, only half number of chromosome is received, each chromosome with double DNA content. After anaphase, the phase is telophase 1. In this phase, reorganization of the chromosome at poles occur to form two haploid nuclei. Nuclear membrane is reformed around the polarized group of chromosome. Nucleolus also reappear and the spindle fiber disappear here. After telophase, the two nuclei formed with half number of chromosome. Cytokinesis. Many times cytokinesis does not occur after meiosis 1, but if it occurs after meiosis 1, the cytoplasm divides at its middle by cell furrow in animal cell and by cell plate formation in plant cell. This produces two daughter cells with half number of chromosome and double DNA content. Generally, there is no interphase between meiosis 1 and meiosis 2. A brief interphase called interkinesis may occur in some cases. There is no duplication of chromosome occurred, only G1 and G2 phase occurred. Meiosis 2 involves in four substage: prophase 2, metaphase 2, anaphase 2 and telophase 2. The second meiotic division is very much similar to mitosis, but the only difference is that the DNA does not duplicate, whereas the centromere duplicate. Meiosis 2 is also called as equational division. The first phase of meiosis 2 is prophase 2. In this phase, chromatids have widely separated arms, nucleolus and nuclear membrane disappear, spindle fibers are formed at each pole. Next to prophase 2 is metaphase 2. Here, the chromosome move to the center of equatorial plane. The centromere attach to the spindle fiber and the centromere divide. After metaphase 2, the cell enter into anaphase. The sister chromatids separate from one another. The sister chromatids are pulled to opposite pole due to constriction of spindle fiber. The chromatids after reaching the pole are called chromosomes. After anaphase 2, cell enter into telophase 2. Here, reconstitution of nuclei takes place. The chromosome begin to uncoil and become thin. Nucleolus and nuclear membrane reappear at each pole. After telophase 2, cytokinesis occurs. In cytokinesis, the daughter cell form two cell, in other words, from two daughter cell of the first meiotic division, four cells are produced, each with haploid set of chromosomes. Significance of meiosis. First, formation of gametes. Meiosis produces gametes for sexual reproduction. Second, formation of spore. Meiosis produces spore in plants for asexual reproduction. Third, maintenance of chromosome number. Meiosis half the chromosome number in the gametes so that fertilization may restore the original diploid number in the zygote. Fourth is introduction of variation. Meiosis provide a chance for the formation of new combination of chromosome. This brings about variation. The new combination are produced by crossing over in prophase 1 or by random distribution of paternal and maternal chromosome between the daughter cell in anaphase 1. Variation help animal and plant breeders to improve the race of useful animal and plants. Meiosis may bring about chromosomal mutation, some of which may be beneficial for the organism. From this lecture, I have completed the topic cell cycle and cell division. Hope this will be beneficial for you. In this adverse situation, stay at home and stay safe. Thank you.